my favorite chapters, chapter 13, Introductory Virology. So we're going to talk primarily about viruses. Viroids and prions, while they're in the title, they're really just footnotes at the end. So this is a great chapter because I feel it helps clear up a lot of misconceptions about viruses that people often don't understand or realize they're making. So, just a little refresher on some of the things we talked about viruses back in the first week of the semester. Viruses go by a number of names. Okay, heard of virus, I'm sure. Maybe you've heard of phage. Okay, phage is synonymous with virus. You want to get more technical, sometimes they're called virions. Okay, all synonymous. Phage, virus, virions, all the same thing. The first major misconception people have about viruses is the fact that they are not alive. Okay, we call them non-living particles. Okay, now, does anybody recall what our cells can do that viruses cannot? Okay, why are they non-living? Yeah, they can't, well, reproduce. But yeah, very close. They cannot reproduce on their own. Okay? No mitosis, no meiosis, no binary fission, nothing sexual, nothing asexual. Viruses are non-living because they cannot reproduce on their own. So, they're obligate intracellular parasites. Okay, so we know obligate, right? They have to have something. In intracellular, referring to the fact that they have to get into healthy cells. In short, viruses use your cells to do the reproducing for them. Okay, and we're going to get into all sorts of details with this, but viruses essentially turn your cells into viral factories. Okay, forcing your cells to pump out lots and lots of copies of that virus. Okay, so all viruses need some kind of living host cell to do this replication process for them. Okay, so they cannot reproduce without a host cell. Next thing people don't often realize, how ridiculously simple viruses are. Okay, so when it comes to overall structure, viruses are basically a protein shell surrounding a nucleic acid core. So some kind of rudimentary genome. So for instance, everyone can draw the flu. Start with a hexagon. And then fill in that hexagon with 8 lines. 1, 2, 3, 4, 5, 6, 7, 8. Behold, influenza. That is about it. So it is a protein shell surrounding a genome made up of 8 fragments of RNA. That's it. That's the flu. Really not too much else to it, as we'll see. Another thing people don't realize. Viruses are equal opportunity infectors. Okay? There are viruses of all 3 domains. We're familiar with the viruses of humans. Okay? Ones we deal with day in, day out. But there are also viruses of bacteria. Okay? And that's what we're going to focus on in this chapter. We're going to focus on bacteriophages. Viruses that exclusively infect bacteria. And there's a good reason for focusing on this. They are the simplest model, so it's a good one to start with. And part of that is because their genome is made up of DNA. Okay, so they obey processes that we can all relate to from recent chapters. And then there are viruses of archaeans as well. Okay, so there are viruses of all 3 domains of life. No one's safe. Okay, so let's look at this structure a little bit more. And like I said, there's not too terribly much to look at here. So let's look at the inside of the virus first. Let's talk about this genome they have, this nucleic acid core. So this is where you do get some variety. And what you find inside of a virus is typically how you classify the virus. Okay, so different viral families are broken into about what we're going to talk about. So, let's start first by asking, is it a DNA virus or an RNA virus? Okay. Some of them have a genome made up of DNA, some have genomes made up of RNA. Alright, it's complicated a bit. DNA or RNA, is it linear or circular DNA or RNA? Okay, so for the flu is made up of 8 linear segments of RNA. Okay, but there are viruses that have circular, what looks almost like plasmids, genomes of DNA or RNA. So, DNA, RNA, linear, circular. Next question. Is that genome double-stranded or single-stranded DNA or RNA? So when we look at bacteriophages, we start with them because they are double-stranded DNA viruses. Okay, so they go through transcription, translation, just like we're used to. They're relatively simple. Now, there are single-stranded DNA viruses. There are single-stranded RNA viruses. There are double-stranded RNA viruses. It's just about mixing and matching and thinking of every combination you can. Okay, so this is where the complexity comes in, is what kind of genome do you have? DNA, RNA, linear, circular, single-stranded, double-stranded. Okay? And when we get to the end of this chapter, I'll illustrate this a little bit better when I introduce you to just a little bit about animal viruses. Okay, so this protein shell, we call this the capsid. And all viruses have a capsid. It's what holds in their nucleic acid core. And this is pretty boring because this capsid is made up of almost always a protein called capsid. So all viruses have capsids made up of this one kind of protein, capsid. Now, they can organize these proteins in different ways, so you get kind of different textures to the viruses, which I'll try to show you later on. Okay, so talking a bit about that capsid and how those proteins are organized, you end up with 3 major shapes. Okay, now there are variations on this, right? There's always rule breakers, but most viruses fall into one of these 3 shapes. So, the most common shape amongst eukaryotic viruses is polyhedral. Okay, and I don't know why Pearson gets all cute and calls it polyhedral. Most other books call it icosahedral. Meaning that these viruses have a 20-sided equilateral geometric shape. So I don't know how many of you are familiar with D20 dice. Okay, so 20-sided dice. That is what they look like. Okay, so what you have there isn't exactly 20-sided. The thing is that when you start adding 20 equilateral faces, they become kind of spherical. I got a few more of these. I'm working with Chris Asthanopoulos over in engineering. He's helping me 3D print viruses. This is the biggest one I have. I got 3 little test ones that I'll bring to the lab if you want to see. But I can't remember the name of this guy. But this is icosahedral. Okay. There are 20 equilateral faces here, but that's so many faces that they do look almost spherical. So that's most animal viruses. Now, the next shape is helical. Okay, now think helix, think coil or spring. So here, that protein is just coiled up. Most of these guys look like filaments, rods. And the nucleic acid core, if you're curious, is coiled within as well. So this is pretty typical for viruses of plants. Okay, the classic and most famous example, the tobacco mosaic virus. Okay, that virus has been very important in genetic modification of plants. Okay, so this is typical of plant viruses, even though in the diagram, that's Ebola. Okay, Ebola is technically a helical virus, but it's kind of a weird one because it has more of a, we call it a shepherd's crook shape. Okay, like a bent cane. So like I said, we're going to focus on bacteriophages. Viruses that only infect bacteria. So what about their shape? Ironically enough, they are complex. So what appears to be like an evil little moon lander, that is what they look like. Okay, they got a lot more going on. I'll talk about the different components later on. Okay, but the short version is, it looks like you take a polyhedral virus and then you jam it together with a helical one. Okay, so there's a polyhedral component, there's a helical component, and we'll talk about the little legs later on. Okay, so these are bacteriophages. And they actually have that shape for very good reason. Okay, so the next thing I want to talk about is additional structures you may find associated with viruses. Now, some textbooks associate this next structure with shape. I don't. Okay, so some viruses are what we call naked, meaning you got your genomic core, you got your protein shell, and then there's no other layers beyond that. Okay, it's just naked. These are the viruses that typically lyse their host cell, meaning they will cause it to burst after infection is over. Now, naked viruses are associated with bacteriophages as well. What we won't get to see too much in this level of course, because I don't get into animal viruses too much, is enveloped viruses. So I'm not actually done drawing the flu here. The flu actually does have one more layer. An envelope. And this envelope is a membrane. It is a good old-fashioned phospholipid bilayer, just like the membranes we've talked about all semester. Here's the problem, though. Viruses can't make membranes. It can replicate its genomic material, it can replicate its capsomere proteins for its shell, but it cannot make a membrane. Where did it get it? From the host. So, looking at flu again. So take a human red blood cell. When that red blood cell is infected with the flu, it is going to very gradually fill up with flu viruses. Now, when those viruses want to get out, they don't lyse the host cell. They don't cause it to burst. Instead, they kind of force their way out. They trick your cells into exocytosis. Now, that's a gen bio term. Right? You talk about endocytosis, bringing stuff into your cells, and then exocytosis is how your cells package things to leave. So here, the virus tricks your cell into packaging it. So the virus pushes its way out, and it takes a portion of the membrane with it. Okay, so this is called viral coating. So this is how enveloped viruses get their membrane. They take it from you. Okay, so this is something we do not see with bacteriophages. Okay, this is something you see typically with just eukaryotic viruses. Okay, so at this point, any questions about shape, structure, envelopes before we move on? All right, now I'll talk about that last little bit about spikes there at the end. I'll come back to that. All right, so more misconceptions to clear up. Sizing up viruses. So they are terribly, terribly small. Okay, and to put things into perspective here, think about this. Viruses infect host cells. So would there ever be a virus that's larger than the host cell it infects? No, they would all have to be smaller than their host cells. And when you put it to scale here, it's amazing how much smaller they can be. So the range is 10 nanometers to 500 nanometers. So most of them you need an electron microscope to visualize. But for instance, on the tinier end, we got polio. Okay, compare polio at 30 nanometers to this human red blood cell at 10,000 nanometers in diameter. Okay, big difference in size there. Okay, even comparing the T4 bacteriophage to E. coli, that's a severe difference in size. Some of them do get a bit on the larger end. Ebola's a bit of a beast, but it's somewhat unusual. And there's another example we'll talk about in a second, the Mimi virus, which they actually took out, which makes me sad. Now, with this diminutive size, with these tiny, tiny viruses, that means there's not a lot of space to pack genetic material. So, to put things into perspective here, we can measure the size of their genomes based on how many genes do you find in a virus. And the range is 4 to 1,000 genes. 1,000 is not fair. Okay, most of them are on the lower end here. So, for instance, the flu. We drew its entire genome. 8 fragments of RNA, each one is a gene. So that means the flu, which has killed millions of people throughout history, does so with 8 whopping genes. Okay, so folks, E. coli has 4,000 genes. Humans have 20,000 genes. The flu has 8. They're the image of efficiency. Polio, 6 genes. Paralyze you for life. So most of them do not have very many genes. Now, the oddballs. And I really ought to put a slide in here for these guys, because more and more is coming out each year about them. In the last decade, we've discovered a newer group of viruses called Pandora viruses. They are enormous. They kind of break the rules of what we understand regarding viruses. The first of which was the Mimi virus. The Mimi virus was found to have 1,000 genes. Okay, that is highly unusual because that is more genes than the least amount of genes you find in the simplest prokaryote. Okay, so it's a virus with more genes than a group of bacteria. That should not be. There was one Pandora virus that was found to have ribosomes. Okay, that also should not be. So we're trying to figure out where these weirdos belong. Okay, are these Pandora viruses something that used to be cells? Are they trying to be cells? Okay, is there a fourth domain of life we need to fit in here? So I mention them as always because I try to be proactive as more research comes out, because this stuff will end up in textbooks and day-to-day conversation. Alright. Oh, this is a image I throw in just to show you the fact that viruses are so tightly packed with DNA because of their tiny size that most of them actually have positive pressure on the walls of the capsid. So if you were to pop one, they would just literally burst forth with their genetic material. Okay. So, next thing we're going to look at is one other external structure. Okay, because there's really not much else to look at in terms of structure for viruses. The next structure is something every single virus has to have. It's not optional. All viruses have spikes. So, spikes are these glycoprotein appendages. And usually these spikes stick out from the capsid. Okay, so it looks kind of like a pincushion. So again, they're glycoproteins. Okay, they're sugary proteins. And spikes are a very personal thing to a virus. The actual chemical makeup of that spike is unique to every type of virus. It's like a fingerprint for them. And this is because spikes do one thing. Or they generally do one thing. They attach to host cells. Right? So viruses can be kind of picky. Okay? This pickiness is because of the chemical nature of their individual spikes. That spike will dictate what host cells they attach to. So the classic example of introducing students to spikes is spikes that you're already familiar with and you don't even know it. So, influenza. Every year, hear about in the media, there's going to be H1N1, H5N9, or H number, N number. Those are the two spikes of the flu, the H and N spikes. Didn't even realize it, you already knew those were spikes. So, what does H and N mean? Well, this is what you don't know. Okay? Well, probably. The H spike is the hemagglutinin spike. The H spike is how the flu recognizes your cells, how it attaches to your cells. Now, the flu is unique, and this is something you see with enveloped viruses. They have a secondary spike, the N or neuraminidase spike. One of these years I'll say that, right? The N spike is how the flu gets out of your cells. Okay, so here, since it's an enveloped virus, it forces its way out. Well, like I said, it tricks your cell into doing this. And it tricks it thanks to that N spike. So, H spike gets it in, N spike gets it out with your membrane. So, we've established H and N. But what about that number? Right? H number, N number. This is kind of like strains with bacteria, genetic variants. The number refers to what version of those spikes you're looking at, what genetic variant. So we've got H spike type 1, N spike type 1, H spike type 5, N spike type 9, and so on and so on. Another interesting thing about those spikes, and why the flu is often kind of something we worry about year to year, is that the flu virus can mix and match these spike types. Right? We worry about species jumps from avian bird flu or swine flu jumping into humans because animals can be afflicted with multiple types of the flu at one time. So, for instance, it's a game of mixing and matching. So say you were infected with H1N1 and H5N9 at the same time. They can make something new. They can make H1N9. They can make H5N1 and so on. So they can mix and match those fragments. And this is where we start worrying about creating new strains or getting species jumps from animals to humans, because they can do this. It's called antigenic shift. Alright, so again, spikes. They are glycoprotein appendages used for attaching to host cells. Okay? That leads us to specificity. So these spikes recognize something on the surface of the host cell. Okay? Typically it's some kind of receptor. But in reality, it could be any structure on the host cell surface. Okay, so in the case of bacteriophages, we have, and these are out of order, one of these years will fix this. In the middle picture here, that's a bacteriophage, which has wrapped those little legs, its spikes, around flagella. So it recognizes those flagellin proteins. Up here in the top image, that is a bunch of bacteriophages binding to pili. Okay, so they're covering the sex pili. They recognize the pilin protein. Down here, the bottom image, this is poor E. coli that has the T4 phage binding to its LPS layer. Okay, so those are three examples. Okay, it could be other things. It could be capsules, slime layers, peptidoglycan. Viruses are very picky about what they bind to on that host cell surface. Another great example to illustrate this pickiness, this specificity. HIV. Right? HIV infection leads to AIDS, where your immune system is gutted out. HIV really, really likes your helper T cells, the brains of your immune system. It's a curious thing. There are all these cells in your body, all these tissues, but it wants helper T cells. Well, that's because your helper T cells have the CD4 receptor. No other cell in your body has CD4. This is it. Just helper T cells. So that's why HIV is so picky. Its spikes will only bind to CD4. And that leads us to host range. Okay, with this specificity, you have to ask yourself how many different organisms or different types of tissues can a given virus infect. Most viruses are pretty picky. Most of them are very specific to a particular cell type or tissue type or organism. Okay, but some, though, are broad in their host range. And these are the scary ones. Okay, these are the ones that can infect all sorts of animals or all sorts of tissues in your body. So one example, the obvious one, rabies. Right? Causes a really nasty central nervous system infection which has a very high mortality rate. So rabies is not something exclusive to humans. Right? Think of all the little critters you're afraid of getting rabies from, right? You go outside, there's a weird looking squirrel, raccoon, possum, bat, cat, dog, you name it. Rabies can infect all sorts of warm-blooded mammals, because they will recognize a receptor common to the central nervous system of most warm-blooded mammals. Another one that's really odd, Ebola. Okay. Ebola typically has really high mortality rates, anywhere from 50 to 90%, depending on the outbreak. Ebola actually starts off as narrow host range. During an initial Ebola infection, it actually wants your white blood cells to start. Okay, so it hitches a ride to your lymphatic system. Once there, the infection spreads. And after a while, Ebola says, screw it, I'm going to infect whatever the hell cells I want. And it becomes so widespread that most folks will die just from severe uncontrolled inflammation, leading to internal bleeding and death. So basically, I think with Ebola, if you show signs and symptoms, you got about 3 days left, tops. Okay, so any questions about the spikes or host cell recognition, Jessica?
Is there any chance that if you get exposed to a virus, it doesn't find the receptor and you won't get the virus?
Happens day in, day out. You're exposed to all sorts of nasty stuff, but basically a race against your immune system. Yeah, your white blood cells are constantly cleaning crap out of your body you don't want to know about. All right. So we're going to start venturing into the replication cycles here in just a second. Okay, so just to introduce you folks, for all viruses, there is one of two things that may happen during an infection. Okay, once they get into their preferred host cell, they can either directly cause disease, active infection, make more viruses, kill the host cell, start anew. What they may also do, though, is that some viruses can go dormant. They enter a latent state. So, chickenpox, herpes viruses, HIV, okay, all those viruses that cause incurable diseases, it's because they have gone dormant in your DNA, and you're stuck with them forever. So, some may undergo active infection, others may go dormant. So, we'll hit on all the details here coming up. Okay, so as we ease into the replication cycles, I want you folks to start thinking about something. So for one, all viruses have genes. Okay, genes that can be transcribed and translated into proteins. So they have traits. Okay, so what traits do all viruses have in common? Okay, so all viruses would have the genes to make a capsid. Right? They all have capsids. They all make capsamere. So therefore, they all have this gene. Let's see what else. They all will have the means of replicating their genomes. Now, this one is a horrifically loaded statement because how viruses go about this is very personal to the virus in question, but they all have the means of getting their genomes copied. And then the third thing that all viruses have in common, they have to have the means of getting in and out of a host cell. Right? So, for instance, all viruses have spikes. Therefore, they all have genes for spikes. Okay? So, they all have these three things in common. They can make capsids, they have the means to replicate their genomes, and they have the means of getting in and out of host cells. How they go about each one of these in detail depends on the virus in question. Okay, so, moving on from all viruses, like I warned you, we're going to focus on bacteriophages. Viruses of bacteria, and again we focus on them because they are double-stranded DNA viruses. Okay? They are somewhat relatable to what we know from the semester. So, eukaryotic viruses get fun names like polio, HIV, rhinovirus, adenovirus, whatever. Bacteriophages often get very boring names. So what we have up there is a virus that falls in the family of the T-even bacteriophages. So see, what does T-even mean? Well, it just means T plus a number that's even. So there's a T2 virus, a T4 virus, a T6, T8, T10, and so on. Actually, I don't know how high up they go, admittedly. There's a different family called T-odd viruses. I bet you can guess how they're numbered, right? So T1, T2, or T3, T5, and so on. Murdering my markers. So what we have here is probably the T4 bacteriophage. That's the one that most textbooks look at. And like I warned you, I was going to talk about the components a bit more. Okay, so capsid is at the top. Okay, it's icosahedral. It is packed with, in this case, double-stranded DNA. Okay, so it's double-stranded DNA. Below that, we have the tail sheath. This is helical. So think of that tail sheath like a spring. Okay, it's really the best way of thinking of it. So think of it like a spring. We have the base plate, which I don't have much to mention there. Now these tail fibers, folks, those are the spikes. So while many viruses have their spikes protruding from the capsid, not bacteriophages. The little legs, the little tail fibers, those are the spikes. The pin is actually of interest to us, and it'll be important in a little bit. The pin is an enzyme called, lysozyme. So, lysozyme is an old friend. Okay, and it's an enzyme we're going to mention a couple times as the semester goes on because it's an enzyme that we make. Okay, it is an enzyme that is used to break down peptidoglycan. So, the other thing I like to clear up with this diagram is the fact that while it is very neat looking, it looks horrifically inorganic. Right? It looks metal and plastic. It's not. It is protein from top to bottom with some DNA shoved in. Okay, so it is organic. So what we have here is some poor E. coli cell that is now under siege by a great deal of T4 bacteriophages. And every single one of them have injected their DNA into the E. coli cell. So think of these guys like evil little syringes. Okay, so we're going to get into the replication cycles one quick second here. But moving on from what all viruses have in common, I want to mention what all bacteriophages have in common. So this is traits that you associate with just bacteriophages, not with, say, polio or HIV. Okay, so when a bacterial cell is infected, the virus will inject its DNA into that bacterial cell. Okay? Again, like evil little syringes. The other is once that DNA has been injected, all of these are empty husks. Okay? The shell, the capsid, remains outside. Okay? Think insects mold. Once that viral DNA, and I say RNA, but I think there's only one example of an RNA bacteriophage. Once that DNA gets into the host cell, two things will happen. One, the genes will be expressed. Transcription, translation. And two, that DNA genome will be copied. Okay? And how it goes about this? It forces the host's machinery to do this. It takes advantage of DNA polymerase, RNA polymerase, ribosomes. And then, and this is really for all viruses technically, those replication cycles, I got to fix that. Life is a dirty, dirty word here. Those replication cycles can be lytic or lysogenic. Okay, so, that leads us into these cycles. So, lytic phages. Okay, these are viruses that, upon infecting their host cell, go through an active infection. Okay, so without taking a break, they get in the host cell, they take it over, they force it to make more viruses, and then they kill the host. It's lytic because you end up lysing the host cell. Break. Now, we'll also get into lysogenic phages. We have a little bit of time for that today. Lysogenic phages are curious because they can do everything a lytic phage can. They can go through an active infection if they so choose, but they have an added bonus. Lysogenic phages have the ability to go dormant. And they do that by putting their DNA into the host's chromosome. So this is a forced mutation. So think transduction from horizontal gene transfer. They are transferring their own viral genome and putting it into the host's genome. And there it becomes a stable inheritable mutation. But the thing is, that's only temporary. Eventually, that virus will reactivate, and it will finish out the lytic cycle. Alright, so we're going to talk about these in more detail. Alright, so looking at the lytic cycle first, we'll start with looking at the overview. 5 basic steps to how a lytic cycle occurs. So, to start, we have attachment. Right? The virus uses its spikes to attach to something on a host cell surface. Next step is penetration. Like an evil little syringe, the virus injects its DNA into that bacterial cell. Now, the most complicated step is biosynthesis. Okay? This is where you get expression of the viral genes, you make copies of the viral genome. Essentially, you want to force the host to make all the little viral components. Because in the fourth step, we get what's called maturation or assembly. Now, like evil little Legos, the host cell is forced to assemble the viruses in a highly organized fashion until the inevitable happens, release. Okay? We break the host cell wall, bursting forth all of these new mature viruses. They get out, they find new host cells to infect, and the cycle begins again. Okay, so let's look at these steps in just a hair more detail. Nothing crazy. So, first step, like I said, is attachment. Okay. Bacterial phages have those tail fibers, right? Those spikes that they use to recognize something, some receptor on the surface of that bacterial cell. Could be peptidoglycan, LPS, flagellin, pillin, something to that effect. So it finds its preferred host. Now, the penetration step is actually wickedly clever. So this is where bacteriophages are very different than animal viruses. Okay? Our cells are just surrounded by a membrane. Bacterial cells are surrounded by peptidoglycan. Okay? Peptidoglycan is really, really tough stuff. Right? I keep using the analogy that peptidoglycan is like stacking and welding chain link fence. So you're a T4 phage. You've landed on that peptidoglycan. How do you get through that? Remember those little fangs, the little pins? I said that's the enzyme lysozyme. So evolution's a curious thing. They use the lysozyme to carve a hole through the peptidoglycan. So I don't know how many are familiar with ice fishing. When you go ice fishing in the winter, you need to bring an auger with you to carve a hole through the ice. Same idea. They auger their way through the peptidoglycan cell wall. So, once they've carved a hole through, like an evil syringe, they mash down on that tail sheath, injecting their DNA. Okay? And then the shell remains outside. So, third step, biosynthesis. So if you folks ever get the chance to take a virology class, I highly recommend it. It's a very great class to take. It's actually a really hard class to teach. And that's because of the third step. Viruses are extremely diverse in how they handle biosynthesis. Okay? How they will express their genes or how they copy their genes. Okay? They get very unique here. But what they do all have in common is that once that viral genome's in the host cell, those genes must be transcribed and translated. So, viral DNA is transcribed into viral mRNA, which is then translated into viral proteins. Now, something kind of neat about viral genes is that while there's not very many of them, they are labeled or expressed in chronological order. They're timed. So, when you talk about viral genes, often you refer to early, middle, and late genes, referring to when they're expressed during infection. So early genes would produce proteins that you need right away during an infection. So for instance, nucleases. Most viruses, or bacteriophages, will destroy the host chromosome. They want to chop it up, basically to prevent the host cell from protecting itself. So nuclease would be needed right away because nuclease gobbles up DNA. Now, a late gene example would be the gene for lysozyme, right? Because in penetration, we use lysozyme to carve our way in. If you make lysozyme right away, I imagine it'd probably be a bad thing, right? Because if you make too much of it, you're going to kill that host cell prematurely, right? Because it breaks down peptidoglycan. But you're going to need that lysozyme later on to get out. So, lysozyme would be produced later on to infection. Okay, I don't mention any middle ones because not really appropriate for this level of course. But typically they are expressed in chronological order. Okay, so again, once that DNA is in there, the genes get expressed, the genome gets copied. How bacteriophages do this? They just trick the host cell into doing it. So they exploit the host DNA polymerases. They take their RNA polymerases for transcription. They use their ribosomes for translation. Now, how they actually trick it into doing that, that's something that's very intimate to every virus. Okay? So, for example, some viruses come with their own sigma factors. So they have their viral sigma factors that say, hey, RNA polymerase, only express these viral proteins. Come to these promoters. So they get really cute. Oh, and the last thing. This is really an unregulated process, right? The virus just wants to make as many components as it can, so it needs a lot of raw material. Where's it going to get that from? From cannibalizing the host cell. They gut it out, they break down all of its components, and they use it all to make more viral components. So, 4-step maturation, often called assembly. Because now the host cell is forced into putting these mature viruses together, one component at a time. And this step is actually highly regulated, very tightly regulated. The virus is put together in a very specific order. So, in the diagram, it's a little easier here. So, for instance, capsid is always assembled first. And as that capsid is produced, it's carefully filled and woven with viral DNA. While that's finishing up, the tail sheath is put together. Capsid and tail sheath are combined, and then the tail fibers and pins are added. Okay? So it's done in a highly organized fashion. So the poor host cell is filling up with mature viruses, which leads us to the inevitable release. Viruses got to get out. So, if lysozyme got us in, lysozyme's going to get us out. So the viruses will then use lysozyme to weaken the host cell wall until it explodes. Viruses get out, and the cycle begins again. So, some other fun things about this, things we can measure. Burst size. So how many viruses can you make during an infection? Well, for bacteriophages, you're looking at about 200 on average. For human cells, that'd be way more. You can also measure how long this takes. So burst time. For bacteriophages, 20 to 40 minutes. Right? And the time we're in here, we go through 2 to 3 lytic cycles. Well, it's pretty quick. Oh, a little video thrown in there too for you to watch if you want. And you can actually watch this happen. So we got here is 3 different viruses. We're looking at plaque assays. So compare T2, T4, and lambda phages. All 3 plates has E. coli. E. coli is sensitive to all of these viruses. So you'll notice that there's these little clearings appearing. Those are plaques. And think of a plaque as the opposite of a colony, right? A colony is a pile of bacterial clones. Each clearing is a bunch of cloned viruses. Okay, because what you're seeing here is the destruction of the bacterial cells. And the plaque gets bigger as the virus spreads. So the other thing to notice here is the fact that the plaques are different sizes. These viruses have different burst times. T2 is the slowest, T4 is slightly faster, lambda is really fast. Okay, so how long it takes these viruses to get through these replication cycles depends on the virus. Some are faster, some are slower. Okay, so we're going to go about 5-6 minutes long here because I want to get us halfway through the lysogenic phages so we don't get too far behind. But any questions about the lytic cycle? Okay, so the next thing we're going to look at is the lysogenic cycle. But before we get into that, we have to talk a little bit about the viruses that are capable of the lysogenic cycle. Lysogenic or temperate phages. So, again, these lysogenic viruses can do anything a lytic phage can. They can go through a lytic cycle, or they have the ability to go dormant. Okay, so how do they go dormant? Well, once their DNA gets into the host cell, so let me flip back and forth here, they will actually force their DNA into the host chromosome. They do so through a very unique type of recombination, site-specific recombination. Right? We talked about homologous recombination before. This is what chromosomes do. They're slightly similar, they switch places. This is a little different. Okay? Site-specific recombination is more precise, and it uses it to integrate the entire virus, or viral DNA, into the host chromosome. So, what do you call a virus that's gone dormant? It's called a prophage. Okay, so if you've ever had chickenpox, you are harboring the varicella zoster virus. It is a prophage in your peripheral nerves, most likely your peripheral nerves. Now, this is a terrible term, but what do you call the cell harboring a prophage? You call it a lysogen, which is kind of backwards to me because you'd think the virus would be called a lysogen, but no, that's what you call the cell just to confuse the shit out of people. But anyways, once it's become a prophage, that virus is a stable inheritable mutation. It's just sitting there in the chromosome, like it's always been there. It gets copied, it gets passed on to future generations, until the inevitable happens. That virus reactivates and finishes out the lytic cycle. Okay, so the duality of lytic phages. So the classic example is phage lambda. So you get attachment and penetration, that's the same. Now, once the virus gets in there, it has to make a decision. Am I going to go through the lytic cycle, making more of myself? Or am I going to become a prophage and go dormant for X amount of time? So if it does go dormant, again, it's basically a mutation. It's copied and passed on to all the other cells, which in turn become lysogens. Until the inevitable, where the virus removes itself from the chromosome and finishes out the lytic cycle. So, the lysogenic cycle is essentially a pause button, as long as nothing goes wrong. Just real quick, how does it get to make this decision, right? How does a virus choose whether or not to go dormant or to immediately kill the host cell? Well, in the case of bacteria, it depends on the state of the host cell. So remember growth curves, right? Lag phase, log phase, stationary death. To become a prophage takes time. Okay, to integrate the viral DNA into the host DNA, the host cell must be growing slowly. So if the phage is going to lysogenize the host cell must be in, say, stationary phase. It's got to be growing slowly. Now, on the other hand, if the host cell is growing quickly, think log phase, then there's no time to become a prophage. Now you got lots of host cells to infect, so they'll go through the lytic cycle. Now, these are not absolutes. Okay, these are just trends. Okay, host cells growing slowly, odds are it'll become a prophage. If the host cells are growing quickly, odds are they'll go through the lytic cycle. This will be the last one I talk about, and then I'll shut up. So, this is one of those diagrams the textbooks are starting to take out. It really bothers me because it helps kind of paint a better picture of what's happening. So, I don't go into horrific details of recombination. That's more for a molecular biology class. But I do like to show you folks the consequences of recombination. So here we have site-specific recombination. So how does a lysogenic phage force its DNA into a host chromosome? Alright, so at the top, we have the circular chromosome of phage lambda. Okay, this is a virus that infects E. coli. So right below that, we have E. coli DNA. Okay, that's this stuff. So here's a really curious thing for you. You've got the ATT site on the virus and the bio site on the E. coli DNA. These two segments of DNA are complementary. In fact, they're identical to each other. So the virus has a little bit of E. coli DNA. E. coli's got a little bit of viral DNA, if you want to think of it that way. So they share these very specific segments of DNA. So when the virus gets into E. coli, those sites align, they crisscross with each other, so you get a crossing over event. But this time it's very precise, and the entire virus integrates its DNA into the host cell. Okay, so it loops in. It becomes a part of E. coli's chromosome. So the last quick thing here to mention is, enzymes are always really obviously named, typically. And I love just how blatantly they name these. So the enzyme that puts the virus into the chromosome, conveniently called integrase, and when the virus wants to leave, because it's time to make more viruses, it does the whole thing in reverse. And that enzyme's called excisase. Why not? You don't have to know the names of the enzymes. I just find that humorous. Okay, so this is where we left off. So when a lysogenic phage becomes a prophage, it must maintain its latent state. And what that entails is repressing its genes. Okay? Prophages have genes, right? This is viral DNA. But in order to remain latent, those genes must be turned off. Okay? How they go about doing this, that's really for a virology class. But the thing is that for many prophages, while most of the genes are repressed, not all of them are. So imagine a prophage does leave a couple of genes on to be transcribed and translated into viral proteins. So it's a curious thing, right? The virus maintains a mostly latent state, but it needs a couple of proteins produced. Why is that? When this occurs, it generally results in what we call phage conversion, meaning those proteins that are still produced by the virus, even though it's dormant, will alter the phenotype of the host. The viral proteins change the behavior of the host cell. And that's kind of weird, right? What sort of ways would it cause the host cell to change? Okay, well, let's look at the obvious. All prophages will prevent superinfection. Okay, and superinfection means they prevent identical viruses from infecting that host cell. Now that really makes sense if you think about it. So imagine you're a prophage, you are dormant in this organism's chromosome. You don't want another virus of the same type coming along and killing that host cell. Right? So effectively, the prophage makes the host cell immune to viruses of the same type. That's pretty handy. But there's a catch. This does not affect unrelated viruses. Okay, so the classic example with E. coli is phage lambda. Okay, phage lambda can put itself in E. coli's chromosome. Okay, so other phage lambdas cannot affect E. coli at that point. But if the bacteriophage T4 shows up, that's a lytic virus, it can't stop it. Okay, T4 could still infect that poor E. coli cell. Okay, so this prevents superinfection of related viruses. Okay, does not influence unrelated viruses. All right, so another example of phage conversion, and this isn't always the case with prophages. This is a sometimes thing. Occasionally you encounter prophages that come equipped with toxin-producing genes. Okay, so for instance, and I'll lean on this example a few times towards the end of this chapter here, our old friend E. coli 0157H7, the infamous jack-in-the-box strain. Okay, this strain of E. coli popped up about 20, 30 years ago. It causes enterohemorrhagic fever in individuals. It is a potentially lethal strain of E. coli. Normally, E. coli, even infectious E. coli, isn't that particularly dangerous, but this one is. Why? It produces the Shiga toxin. Normally, E. coli cannot make the Shiga toxin. Its cousin, Shigella, does, though. Okay, so how did this strain of E. coli come about, this gene? Well, it harbors a prophage that took the gene from Shigella. That's transduction. So, there's a number of other pathogens you'd be surprised by that can only produce toxins because of a prophage that permits it. So, diphtheria, botulism, certain strains of salmonella, cholera. Okay, all of these organisms are pathogenic because they just happen to harbor a prophage that allows them to produce a toxin. Now, another nasty one is Strep pyogenes. Okay, this organism causes strep throat, which on a good day, normally strep is highly virulent, but certain strains can cause scarlet fever. So imagine strep throat with the added bonus of having a scaly rash across your torso and a very high fever. These strains of strep harbor a prophage that makes what's called a superantigen, a toxin we'll talk about in a later chapter. Okay, let's see. And then the last example I have for you of phage conversion is they will make proteins that maintain their latent state. Now, this is a loaded statement because these proteins can repress the genes on their own, or they can influence the host cell to aid in repressing the viral genes. That's why it falls under phage conversion, because it can influence the host to help out. So, those are my 3 examples I have here of phage conversion. There are others, but they can prevent superinfection of related viruses, they can produce toxins, and they can influence the host in helping maintain the latent prophage state. But any questions about lysogenic cycle, lytic cycle, phage conversion at this point? Okay. All right, so next question to ponder upon. So this prophage state is supposed to be temporary. It becomes a part of the chromosome for X amount of time, but eventually the inevitable happens. That virus is going to remove itself from the chromosome, and it's going to finish out the replication cycle. It's going to make more of itself, killing the host. So we've kind of addressed how it chooses when to go dormant, but how does it choose when to reactivate? Well, in short, 1, it's still not well understood why prophages reactivate. But 2, it's often spontaneous. Okay? Just random chance the virus reactivates. The only thing we do understand, though, that could cause a prophage to say, I need to get the hell out of here, is when a host cell becomes stressed, old, or damaged. So it's not that the prophage says, oh, well, this cell is no good, I need to get out of here. It's just the prophage is no longer as likely to be repressed if the host cell is growing old. So, for instance, chickenpox. Right? My audience grows younger as I grow older, and less of you experience chickenpox. But for those of you who have, you harbor the varicella zoster virus. It is in your peripheral nerves. So, as a kid, you get chickenpox, you get through it. It's not particularly awful, just really itchy. But then, 40 to 60 years later, what happens? It comes back as shingles. Same virus. But the thing is, think of the demographic that gets shingles. It's generally the elderly, right? Your nerve cells are not really helping in maintaining that virus in a repressed state. But, again, that's just where it's more likely. There's nothing to say that you couldn't get shingles in your 20s. Okay? This can be totally spontaneous. So, prophages reactivate either A, just through spontaneous chance, or B, because host cells are growing weaker or older. Okay? They're just not maintaining that repressed state as well. All right, so this is a slide I meant to end on last time. See how far behind I am. So again, the duality of lysogenic phages. Okay, they can either go through the lytic cycle if they so choose, or they can become dormant as a prophage, where this is just temporary because eventually they will finish out the lytic cycle, making more of themselves. There is an animation you can watch for study purposes if you want. Okay, so that's it for the replication cycles. And all viruses undergo these replication cycles. Just how they go about it can vary wildly. All right, so that leads us to our first loose end. Transduction. Now, we've already talked about transduction. We've only half talked about it, though. Okay? So this was something we talked about back in Chapter 8b with horizontal gene transfer. So we had 3 mechanisms where bacterial DNA can be transferred from one bacteria to another. Right? We had transformation, the uptake of naked DNA by a cell. We had conjugation, where the cells come together and make direct contact. And then we had transduction, where bacterial DNA is moved from donor to recipient by a virus. Okay, we talked about generalized, which I'll refresh over again here in a second. But we have not talked about specialized. This is why. Specialized transduction is the result of the lysogenic cycle. And that would have made no damn sense if I tried to talk about it a couple chapters ago. Okay, so, refresher on generalized transduction. So, this is the movement of DNA from one bacterial cell to another by a lytic phage. Okay, so this is the consequence of a lytic cycle. And again, this is where something goes wrong during the lytic cycle. Okay, so we've gone through the steps, right? You get attachment, penetration, and then during biosynthesis, these lytic viruses will usually chop up the host chromosome. Chop it up to as many little pieces as they can. Now, the mistake occurs at step 3 here, during assembly. So when viruses are normally assembled, as we saw last time, it's a very precise process. Right? The viral capsid is assembled and very carefully packed with viral DNA. But nothing in nature is perfect. Okay? Mistakes can happen. And the mistake here occurs when you get a virus that is packed with bacterial DNA accidentally and randomly. So, you get a dud virus. Okay, so most of these are perfectly viable viruses. You get a one dud here. Then you get to release. Right? The viruses get out. Okay, so the thing is that the capsid is still intact. This guy still has spikes. So it attaches to another bacterial cell. So you get attachment, penetration, but this time we are injecting it with bacterial DNA. Okay, the DNA that was mistakenly packaged. And then if you get recombination, you get a stable heritable mutation. Okay? So, this is generalized because any piece of that chromosomal DNA could have been packaged. It's random. Okay, so any gene from that donor cell could have been transduced, could have been moved to the recipient cell. Okay, so this is a consequence of the lytic cycle, okay, because it happens during assembly of the lytic phages. All right, so what we haven't seen is specialized transduction. And again, like I mentioned, specialized transduction occurs when there's an error during the lysogenic cycle. So this involves prophages. Something goes wrong with the prophage. So I'll bounce back and forth here. So here's that diagram for site-specific recombination I love so much. Okay, and this will illustrate nicely, at least I hope it will, where the mistake happens. So normally, you get your viral chromosome. It will recombine and loop into the host chromosome where it becomes a prophage. Okay. So all that happens normally. The mistake happens when the virus leaves. Okay. Normally, when a prophage reactivates, it does all this in reverse. Okay. It loops back out precisely, so just the virus leaves. The mistake happens when it overshoots its mark, when it takes a little extra DNA with it. So these are the possible consequences. So normally, you would just get phage lambda leaving with phage lambda DNA. But if transduction has occurred, this is the two possibilities. So, notice on the prophage at the bottom, the bacterial DNA to the right and left is blue and red. Okay, think of those as two different genes. Blue gene, red gene. So the virus could leave taking some of the blue DNA with it. Or, gene overshoots the other way and takes some red DNA with it. Okay, so this is the mistake. Okay, normally just the virus leaves. But in the case of specialized transduction, it could take adjacent DNA with it as it leaves. So it just overshoots the mark. Takes some extra DNA with it. So this is why it's specialized. Okay? Not any old gene is going to be transferred. Just the genes that are potentially adjacent to the prophage, whatever's to the right or left of it. That is the only DNA that can be transferred. So it's hard to see in this diagram here. They made it too small. But here they're showing you that the gal gene was transferred. So you get a little spot of purple here. Okay, so it took this extra bacterial gene with it. So it finishes out the lytic cycle. So all the new viruses, they get packaged with copies of this kind of faulty viral DNA with the extra gene attached to it. They go out, they find new host cells, they inject that DNA and say they become a prophage again. And look at that. They transferred that gene from the donor to the new recipient cell. So let's use another example we can relate to. E. coli 0157H7. It has a prophage. It's the recipient. That prophage has the Shiga toxin gene. Okay? Normally, E. coli does not have the gene for the Shiga toxin. What does? Shigella, E. coli's cousin. This is probably what happened to Shigella. It had a prophage. The prophage took the Shiga toxin gene when it left, and it then infected E. coli and introduced the Shiga toxin to E. coli. So, there is a nice example of specialized transduction that you can relate to. Okay? We transferred the Shiga toxin gene from Shigella to E. coli by a prophage. Alright, so, any questions about specialized transduction? Alright, and generally, these guys are still perfectly viable, so this prophage can just keep transferring that gene from cell to cell to cell. Okay, it's not like a lytic phage where this guy was a dud and this is a one-shot deal. These guys can keep passing, which is kind of dangerous. Okay, so the next loose end, and this is where I say we're done with bacteriophages. Okay, so when we talked about viral structure, okay, we said viruses all have that capsid capsid, and within that capsid, you had some sort of genomic core. We mentioned it could be DNA or RNA, single-stranded, double-stranded, linear, circular. What I was alluding to was this. There are 7 major viral groups. Groups 1 through 7, defined by what sort of genomic core they have. So here's the sobering thing. 7 groups, we have only talked about group 1. All this time, we have focused on just double-stranded DNA viruses, bacterial phages. And I'll give you a couple of other examples here in a second, but this is what's appropriate for this level of course. If you take virology, what they do is, as the 16 weeks go on, they break down each group one by one. And it's basically just wash, rinse, repeat. Okay? Talk about the lytic cycle. Talk about the lysogenic cycle if it's there. See how each group goes about those cycles in their own way. Okay? So virology is a lot of repetition. It's a lot of molecular biology, to be honest. So let's see, what do we have here? See, group 2, single-stranded DNA viruses. Group 3, double-stranded RNA. They play by weird rules. Group 4 and 5, they're both single-stranded RNA, but one's positive sense and one's minus sense. I'll show you that here in 1 second. And then group 6 and 7, those are the nasty retroviruses. That's when you get into chickenpox, HIV, and herpes viruses, the incurable ones. So, another way of illustrating these groups, you don't have to memorize this. This is just to kind of help folks understand what the hell's going on with those groups. An important question to ask yourself when you go through the chapters in a virology class is, with each group, the question is, how do you get to mRNA? Okay, so for instance, right, we saw group 1, double-stranded DNA viruses. They have genes. You just transcribe the genes into mRNA. We get that. But what about, for instance, a minus-sense single-stranded RNA virus? It's already RNA. You don't have to transcribe anything, right? These guys are actually mRNA ready to go. So imagine there's no DNA involved. It is mRNA that's just ready to be translated immediately. Make it a little weirder. Positive sense single-stranded RNA viruses. They're actually complementary copies of mRNA. So they have to be turned from RNA into complementary mRNA. So how does that happen? Well, imagine this. There's an enzyme that these viruses produce called RNA-dependent RNA polymerase. It is an RNA polymerase that reads RNA and turns it into RNA. Right? When we talked about RNA polymerase, it read DNA and copied in RNA. Okay? These viruses don't play by the rules that we're used to. So this is a couple of examples. But the big question is, with all these groups, is how do you get to mRNA? All right, so I'll have a couple of examples here in a second. But, like I mentioned, we have only focused on bacteriophages. They're simple, they're straightforward, they're all double-stranded DNA viruses. Well, what about viruses of animals, right? These are the ones that we can relate to. These are the ones we know about. Here's the problem with animal viruses. They're ridiculously diverse. There are examples from all 7 groups. From double-stranded DNA to single-stranded RNA retroviruses. So, you want to get into detail with those, you would probably have to take a virology class to really understand them. But I can introduce you to some of the differences that animal viruses have opposed to bacteriophages. So, for instance, how do many animal viruses get in and out of animal cells? Okay, well, that can be a little different because our cells don't have a cell wall, right? It's just squishy membranes. So some animal viruses are capable of just punching right through the membrane. Okay? Polio does this. Polio will just pop a small hole in your cell membranes. But many of them, though, they have to be engulfed by your cell, swallowed up, packaged through endocytosis. And then how do they get out? Okay? Some of them just punch their way out of your cells, like bacteriophages. But others, though, have to be packaged up. So they have to be shipped out via exocytosis, which I'll show you in just a minute. So I have a couple of videos here that illustrate that. Okay, those major differences, how they get in and how they get out. Now, when it comes to actual replication cycles, it doesn't matter. All 7 groups, attachment, penetration, biosynthesis, maturation, release. That never changes. It's just how they go about each step will vary immensely, especially biosynthesis. So, to illustrate how ridiculously complex it is, I included a video on HIV. I pray the link still works. I forgot to check it again today. But I'll also have a diagram to show you here in just a second. Okay, so just a couple of quick examples of animal viruses to show you how they differ from bacteriophages. So the first one I got, influenza. Okay, so the flu. Instead of carving their way into your cells, flu viral particles have to be packaged up by your cells. So imagine you have, they have epithelial cell here, it could be red blood cell, whatever. So you can see it's being packaged up. Okay, so that's endocytosis. Once it gets packaged up, you can see it's now in a vessel. And then, when you get to step 3, you get what's called uncoating. So you can see the virus, it looks like it's vomiting out its contents. That's exactly what happens. the virus gets uncoded and empties out its 8 RNA segment genome. Okay, so the flu is an RNA virus. Alright, so you get to biosynthesis. So the flu is one of those positive sense RNA viruses. So the RNA has to be copied into RNA. So I mentioned that RNA-dependent RNA polymerase. The flu uses that. Okay, so the flu's genome or fragments are copied, the flu genes are expressed, and you get to assembly. Right? New flu particles are assembled, and then you get to release, where, as you can see, they have to be packaged up through exocytosis. And they just kind of bubble out and leave one at a time. So, that's a little different from what we saw with bacteriophages. So for one, it has to be engulfed by your cells. 2, it's an RNA virus. So in this case, its RNA has to be copied into RNA. And then during release, it has to be packaged up again. And then this is where things get stupidly messy and complex. HIV is a retrovirus. And retroviruses are lysogenic, meaning if you get them, they will put their DNA into yours and they become a new little friend for the rest of your life, right? If you've had chickenpox once, you are now harboring chickenpox for the rest of your life. The virus is dormant within you. It's the same with HIV. So, and I'm just hitting on some points here. This is very complex stuff. But, so you get the HIV virus, you can see it's enveloped. It is targeting a helper T cell. So penetration this time actually involves the virus merging its membrane with your own. So once it merges, it can dump in its viral genome. So HIV is another RNA virus. But unlike the flu, HIV will actually copy its viral RNA into viral DNA. So when we talked about gene cloning, we went through that awful slide on how to clone human genes, and it talked about reverse transcription. You take the mRNA and turn it back into cDNA. This is the virus we stole that from. Okay. The flu uses reverse transcriptase to convert its RNA genome into a DNA genome. And it does that because it needs that DNA genome to force into your DNA chromosome. Okay, so this is an RNA virus that turns itself back into DNA just so it can become a prophage. And now you are stuck with HIV in your helper T cells and you're not getting rid of it. So unlike the lysogenic phages we're used to, as a prophage, it will actually continue out the replication cycle. Okay, so it's only really kind of half dormant. So it continues making copies of its viral RNA, viral proteins, and then you get to assembly and release, where the viruses get packaged up by your cells, sent out, so they can infect other helper T cells. Okay, slowly killing them one helper T cell at a time. So there's a lot of differences here. Right? This is way different than the lysogenic phages we saw before. Alright, so any questions with the flu or HIV? I'm just hitting on really minor points with these. I'm not going to ask anything in great detail regarding them. Okay, bacteriophages, though, I can ask anything I want. Alright, so last two quick loose ends here. Viroids and prions. And while they are part of the title for the chapter, they're really footnotes because short version is they're not well understood. Okay, viroids we know even less than prions. So imagine this: a viroid is an infectious piece of RNA. Okay, a little piece of RNA gone bad. Okay, there's There's no protein shell. There's no genes. It's a little chunk of RNA that's become infectious. This is weird to me and weird to scientists because viruses make sense. Viruses have rules. They've got the shell, they've got spikes, they have different genomes, but this makes no damn sense. Okay, so the things we do know. Viroids are small, single-strand pieces of RNA. They're about 300 some odd nucleotides long. That's a problem. They are smaller than the smallest known gene. They do not contain genes. Okay, so it's a 350-ish nucleotide stretch gone bad. They like plants, okay, especially plants that make tubers like potatoes. And when they do infect these plants, they cause spindle tumor viroid syndromes. So you can see at the bottom here, healthy potatoes versus two other types of potatoes that were infected by two different types of viroids. Other things we know, they start off in a linear state, but they become circular for some reason. But that's about it. Right? We do not know how they replicate. We do not know how they cause disease. We do not know how they get from plant to plant. And we do not know of any other examples outside of plants. This is it. You know about as much as I do. Prions are a little bit more well understood. There's actually a lot more research into these guys. So prions are infectious proteins. So there's no DNA or RNA involved. This is quite literally a protein gone rogue. And that's the best word for it. They've gone rogue. So prions cause spongiform encephalopathy. They cause neurological decay. And they cause this in a number of warm-blooded animals. So I guarantee you've all heard of mad cow disease. Right? That is a prion-related disease. That's spongiform encephalopathy. We discovered it in sheep. Sheep, it's called scrapie. In humans, it's called Creutzfeldt-Jakob disease, so CJD. So here you can see normal, healthy brain tissue. There's little neurons there. And then with the prions, you get these spongiform lesions that appear. Okay, so your brain becomes quite porous, and you die from neurological decay. So what's going on in the brain? Why is this protein so dangerous and deadly? Well, the funny thing is, like I mentioned, it is a rogue protein. It has a healthy counterpart that's found in every single one of your brains. So here's a prion, PRP protein. Here's its healthy counterpart, PRP. The difference between them, the prion has been misfolded. It has taken a different shape. When the prion gets into your brain, what it does is it finds its healthy counterpart and misfolds it, turning it also into a prion. And then they find more healthy counterparts. And they continue this cascade of misfolding. So this is a bit of an exponential disease. As they continue to make more of themselves, it spreads like crazy. Until all you're left with is the misfolded version of the protein. So these guys are still kind of weird because we don't know where they come from. We don't know how they're spread. We do know they're really damn durable. We know that in many cases prions can survive autoclaves. So we've taken a long, hard look at surgical sterilization protocols because we know prions can survive a lot of our sterilization techniques. Do know they won't survive fire, so they can die from that. Or die. But the only other kind of neat thing I have to mention is there's been a lot of researchers that have been trying to compare prions to Alzheimer's because the way that Alzheimer's progresses and pathology looks very much prion-like. The way lesions and plaques